Dear students, hello and assalamu alaikum. I'm Ali Abbas Kazalbash, and today's program will be on the study of hereditary materials known as nucleic acids and on the process of protein synthesis. Now, nucleic acids are the essential components of life. They are found in the chromosomes in the nucleus of all living cells, and they code for all the functions that are carried out by each living cell and organism. Nucleic acids are then passed on from one generation to the next through the process of replication and reproduction. It is in these nucleic acids that determine the morphological and physiological characteristics of an individual. It's the sequence of nucleotides on the nucleic acids that code for amino acids that make the specific molecules of protein in order to perform the desired functions. Now, with the dawn of the new millennium, scientists have now mapped the human genome, which is actually the code of nucleotides held within the nucleic acids, in order to better understand the process of hereditary diseases and to ascertain a cure for these ailments. This further amplifies the importance of nucleic acids and the process of protein synthesis, which is directly linked to the sequence of nucleotides found on the nucleic acid. Today's program is dedicated to the structure of nucleic acids and the process of protein synthesis. Now, basically, there are two main types of nucleic acids: deoxyribonucleic acid, DNA, and ribonucleic acid, RNA. Now, deoxyribonucleic acid, or DNA as it's more commonly known, is a complex molecule composed of several repeating units. Now, as there are four kinds of nitrogen bases, there are also four types of nucleotides. Each nucleotide is named upon its base. These nucleotides are linked to form a polynucleotide chain. In this linkage, the sugar of one nucleotide is bound to the phosphate group of another nucleotide, and so on. In every nucleotide, the phosphate group is attached to the fifth carbon of the sugar, and during the process of linkage, the phosphate group of one nucleotide is linked to the third carbon of another nucleotide. Now, after the chemical analysis of DNA, attempts were made to know as to how these components are arranged on the DNA molecule. Now, the first attempts were made by Wilkins and Franklin. They used X-rays for obtaining the three-dimensional picture of a DNA molecule. They found out that DNA is a helical structure and that there are at least two helices coiled around each other. The salient features of the Watson and Crick model are: each DNA molecule is made up of two chains or strands, which are coiled together in a twisted form looking like a coiled ladder. Now, the upright portion of this model consists of the sugar and phosphate nucleotides, whereas the rungs are made up of the nitrogen-based nucleotides. Each rung consists of a purine linked with a pyrimidine. That means adenine will be linked with thymine and guanine with cytosine. Now, both the nucleotide chains are attached in an opposite direction, as visible in this model. In other words, a 3' end and a 5' end. Now, both the chains are held by weak hydrogen bonds, which you can see over here. Between guanine and cytosine, there are 3 hydrogen bonds, whereas between adenine and thymine, there are 2 hydrogen bonds. The amount of adenine in one strand is equal to that of thymine on the other, and the same is the case for guanine and cytosine. Now, dear students, the Watson and Crick model of DNA did not only explain its chemical structure, but also the mechanism by which it duplicates itself. When the synthesis of DNA starts, both the strands are separated due to the breakage of the hydrogen bonds. Then, one strand is cut and resealed after releasing the tension of the DNA molecule. The separation of both the strands and the release of tension is done by helix destabilizing proteins and enzymes called topoisomerases. Now, both strands, after detachment, act as a template for the synthesis of new strands. The new strands are synthesized by enzyme DNA polymerase. The DNA polymerase, taking the original strand as a template, adds complementary nucleotides to synthesize the new strand. In other words, if the old strand has TGG as its nucleotide sequence, then the DNA polymerase will add ACC to the new strand or complementary strand. In this way, 2 daughter molecules of DNA are produced from one DNA molecule. In each daughter DNA molecule, one strand is the original and the other is the newly formed strand. This means that the DNA replication is said to be semi-conservative. The enzyme DNA polymerase adds nucleotides in only the 5' to 3' direction. As the DNA molecule has 2 antiparallel strands, thus the enzyme carries on continuous replication from the 3' to 5' end. But on the other strand, in other words, the 5' to 3' strand, the replication is discontinuous. Here it occurs in the form of small fragments, and these fragments are later on joined together by the enzyme DNA ligase. The replication of DNA starts from certain special sites called replication insertion sites, RIS. And on a long DNA molecule, there may be several RISs. Dear students, now before going into the process of protein synthesis, we must first be familiar with the genetic code. Actually, a particular gene controls the synthesis of polypeptide chains, and the gene is nothing more than the sequence of nucleotides or bases along the length of the DNA molecule. There are only 4 types of bases, but 20 kinds of amino acids. Now, if one base codes for one amino acid, then 16 amino acids remain without codes. If 2 bases code for one amino acid, then there will be 16 codes, and again, 4 amino acids have no codes. If 3 bases code for one amino acid, then the codes are sufficient for 20 amino acids. This is the reason why the genetic code is called the triplet code. Now, the genetic code is universal, except in the case for the mitochondrial code. In other words, in all living organisms, one code, for example, UCA, spells a specific amino acid, and that's serine. Now, this code system is having no commas or full stops. Therefore, the insertion or deletion of any base changes the whole sequence of amino acids and onwards. The examples of codes shown here indicate that the 3rd base is not so important in some codes, and in some the 2nd one. However, in most of the codes, change in the 1st base changes the code and hence the amino acid and protein as well. the change of one single amino acid may cause the failure of protein function. For example, in sickle cell anemia, glutamate is replaced by valine, which results in sickle cell anemia. The cell organelles responsible for protein synthesis are basically three types of ribonucleic acids, RNAs. These RNAs have the same composition as does DNA, except in the case of RNA, there's a ribose sugar instead of the deoxyribose sugar in DNA, and the uracil nitrogen base instead of the thymine nitrogen base. Now, there are three types of RNA molecules: ribosomal RNA, rRNA, messenger RNA, mRNA, and transfer RNA, tRNA. The ribosomal RNAs combines with the proteins to form a granular structure called ribosome. These ribosomes are present in the cytoplasm as well as being attached on the rough endoplasmic reticulum in the cell cytoplasm. Now, the ribosomes exist basically in two subunits. The smaller is 40S and the larger of 60S. Upon combining, both these subunits give rise to a complete ribosomal particle of 80S instead of 100S. This is due to the fact that when biochemical reactions occur between two molecules, certain things are released to make the bondage. The unit S stands for the scientist Stredberg, specifying the sedimentation rate of a specific particle or molecule in the medium during ultracentrifugation. Now, the large subunit of a ribosome has basically two sites: the aminoacyl site, designated as A, and the peptidyl site, designated as capital P. The P site has an enzyme peptide synthetase on it. Now, the messenger RNA, the mRNA molecule, is produced inside the nucleus by an enzyme RNA polymerase. Both strands of the DNA molecule open up, and the RNA polymerase copies the coding strand sequence of nucleotides. The RNA polymerase reads a specific DNA sequence and adds the complementary nucleotides to produce the messenger RNA. For instance, if the sequence of nucleotides in a coding strand of DNA is, let's say, TACGGATT, as given here, then the complementary nucleotides on the messenger RNA will be AUGCCCUAA. In all cases of RNAs, the uracil U is incorporated instead of thymine, the T part of the nitrogen bases. The primary messenger RNA is immature and long. It does not enter the cytoplasm in this form. Only after the extra segments are removed, the mature form of the messenger RNA is produced, which then leaves the nucleus and enters the cytoplasm. Now, the messenger RNA has 1. Ribosomal binding sites that contains the AUG triplet codon, which is called the starting or initiation codon. Secondly, it contains amino acids coding codons. And thirdly, it contains a termination codon, UAA, present at the end of the messenger RNA. Mostly, the messenger RNAs have a length of about 900 to 9,000 nucleotides, coding for 300 to 3,000 amino acids. Now, transfer RNA basically acts as the laborer. It picks up the amino acid and carries it to the ribosome that is attached with the messenger RNA. For each kind of amino acid, therefore, there is a particular type of transfer RNA, or tRNA. So there are at least 20 tRNAs present in the cytoplasm, each one for a particular amino acid. Now on the tRNA, there are 4 sites. An anticodon site, which is matched with the codon of the messenger RNA, the ribosomal binding site, the enzyme recognition site for the enzyme that activates the amino acid before binding with the transfer RNA, and the amino acid binding site. Now, amino acids are the raw materials for protein synthesis and are large organic molecules containing carbon, hydrogen, oxygen, and nitrogen. There are, as I said previously, a total of 20 amino acids found in nature, and plants synthesize this amino acid, and animals have to depend upon the plants for obtaining the amino acids in the form of plant proteins. Now, all amino acids are similar in having a hydrogen atom, an amino group, and a carboxyl group. But they differ in their reactant, or R groups. Now, all of these 4 groups are attached to the same carbon, which is called the alpha carbon. The amino acids are linked with each other by a peptide bond. The peptide bond is formed by an enzyme called peptide synthetase, present on the large ribosomal subunit. Now, for each peptide bond, 1 molecule of water is released. But during protein digestion, the enzyme utilizes that 1 molecule of water to break the peptide bond. So this is basically a reversible reaction. Now, assemblage of amino acids into proteins, or the process of protein synthesis, is referred to as the process of translation. The organelles responsible for the synthesis of proteins are produced within the nucleus. Then these come out into the cytoplasm through small nuclear pores present in the nuclear membrane. The message on the messenger RNA is in the language of codes. This code language is translated into a polypeptide chain by ribosomes. A sequence of these nucleotides, the length of the DNA or messenger RNA, is then called the genetic code, and it represents a single amino acid. Now, any change in the sequence of nucleotides changes the code and hence the amino acid at the protein level. And the process of protein synthesis is completed in 3 steps: the chain initiation, chain elongation, and chain termination. Now, firstly, a special transfer RNA, tRNA, is loaded with the methionine amino acid, the initiation codon. This then binds with a small ribosomal subunit, the 40S unit. This complex binds with the messenger RNA in the ribosomal binding site. After this, the large ribosomal subunit, the 60S, binds with it. Later on, the first methionine amino acid is removed from the polypeptide chain. Now, the chain elongation starts soon after the binding of the large ribosomal subunit with the messenger RNA. A tRNA loaded with an amino acid comes to the ribosome. It binds with the amino acyl site and matches its anticodon with that of the messenger RNA. If both the codon and the anticodon are complementary, in other words, they match, then the amino acid is handed over to the ribosome. Simultaneously, upon matching the codon, the ribosome moves to the next codon on the messenger RNA. Due to this movement, the first transfer RNA attached with the P site is released. Now, the P site is occupied by the second transfer RNA, and site A, the amino acyl site, is left vacant to accept another loaded tRNA. Again, a new loaded tRNA comes and attaches itself with the amino acyl site, matches its anticodon and hands over the amino acid to the ribosome. In this way, a long chain of amino acids is synthesized. The peptide synthetase forms the peptide bond between the amino group of the first and the carboxylic group of the second amino acid. Thus, the direction of protein synthesis is from the NH2 part to the COOH part, the amino part to the carboxylic part. Protein synthesis is a very fast process, this must be noted, and that approximately 150 amino acids are incorporated in about 1 minute. At the end of the messenger RNA, there is the termination codon, UAA. This codon does not code for any amino acid. Now, when the ribosome reaches this codon, both the subunits split up and the polypeptide chain is released. Now, in some cases, as the ribosome moves forward on the messenger RNA, another ribosome comes and attaches with the same messenger RNA. This process is repeated several times, and as a result, you have many ribosomes attached to the same messenger RNA. This structure is called a polysome or polyribosome. And the advantage of a polysome or polyribosome is that it provides many copies of the same protein molecule simultaneously. To summarize the process of protein synthesis, let's look at the schematic provided. Now, the process is initiated in the nucleus, where the DNA uncoils at a specific site and the desired genetic code is transcribed onto a messenger RNA. Then the unwanted segments of the immature RNA are then cleaved, leaving a mature messenger RNA. Then this mature messenger RNA leaves the nucleus and enters the cytoplasm. At the same time, ribosomal RNA subunits and transfer RNAs are produced within the nucleus and leave the nucleus via the nuclear pores to enter the cytoplasm. In the cytoplasm, there is a convergence of the RNAs within the ribosomal unit. Now here, the messenger RNA is read and translated into various amino acids, which then eventually produce protein molecules after binding with each other, forming the polypeptide chains. Now, these protein molecules are either utilized within the cell or released to produce the desired effect. With this, dear students, we've come to the end of our program. I would like to thank you for your attention. Until we meet again, wish you the very best. Allah Hafiz.